Thank you. So I do understand a little bit of Spanish, but I do not speak Spanish well. So I will try to speak clearly in English to get across the work that I've been doing. This metagenomic analysis is something that's new to me. I have only been doing this for a few years, and I've been enjoying exploring the kind of questions that you can ask through this type of analysis. The work that I'm presenting, I've done with two undergraduate students at New Paltz, Allison and Lola, who have contributed different aspects to this project. And the work is based on a wildfire that's pictured here that occurred in 2002, a few years ago, and really was inspired by Allison, who was very interested in bacteria that survive in extreme environments. So we're, I guess, lucky that there was a wildfire nearby. Try that. It's SUNY New Paltz. This is a picture of the campus where I work. We are a public university, and mostly it's undergraduate students. We do not have any master's degrees or PhD programs in biology. So the work I do is with undergraduates, and it's a little bit covered. Oh, but now we can the wrong thing. There's a beautiful ridge, we call it, a small mountain that we get to see. It's not as beautiful as the bay here, but it's still quite lovely. I know this group of people has a good understanding of the role of bacteria in the environment, so I won't spend a long time talking about that, but I did want to talk a little bit about bacteria in soil. And in these images, we see there's a great variety of activities. Many of the activities are related to supporting plants, right? There's these interactions between bacteria and plants, bacteria in the soil. And the second image is focusing on one particular species that's known to do a lot to help with plants. What needs to be done? Yeah. There's a great diversity of bacteria in soil. There's expected to be somewhere on the long lines of 60,000 different species of bacteria in soil, and a large number of them, depending on the soil type, somewhere between 10 to the 6 to 10 to the 11, 10 to the 10th, bacteria per gram of soil. Another aspect of this talk is appreciation of the issue of wildfires. And the data in these charts is from the United States, where we see that over the years there's been more wildfires, and not only that, those wildfires are burning more land. And I know that you here are pretty familiar with the issue of wildfires. Isn't just last year, there are significant wildfires in the area. So when the soil is burned, it affects the communities of bacteria that are present. Some of the things that change is the loss of the organic matter through combustion. The soil doesn't hold water as well, and there's a release of inorganic nitrogen. These and other factors change the community of bacteria that are present in the soil after a wildfire. There are these, I think I might stand over here. I keep doing that, I apologize. Pyrogenic organic matter, these unusual compounds that are available for bacteria to eat. And bacteria and fungi that love these combustion products are called pyrophilous. Pyro is Latin for fire, phyllis loving, the bacteria that love fire, right? And one reason to look at this is that it's potentially going to be useful to restore vegetation after a wildfire by helping that along with restoring the right kind of microbial community. So we try to learn about this. So the research site, well, what direction is north? Like if you go about, I think, 8,000 kilometers in that way, you'll hit New York State in the United States up here. And a map of New York State, we'd see New York City. Many people are familiar with that, right? And if you go up the Hudson River to here, you hit the New Paltz and the area for this wildfire study. The wildfire happened in a state park called Minnewaska State Park, very popular tourist attraction, and it has a lot of rare species, a variety types of animals and plants, and these ecological communities that make it pretty special. This waterfall is a big tourist attraction, and I also included it. I don't know if this is true, but there's a TV show on Apple TV called Severance. Yeah, that's Woe's Hollow. So yeah, I've hiked to Woe's Hollow many times in my life. So here, New Paltz, Woe's Hollow is about here, and then where the wildfire occurred was on the other side of that ridge, and an area called Napanock Point. It's about 20 kilometers away, so quite close. I'm trying to remember the order. I think that's the right order. No, I guess that's it. So these are some pictures I took in November of 2023, a little more than a year after the fire. And we can see that the trees were burned quite high. I was lucky enough to convince one of my colleagues who studies wildfires to go on this hike with me. And he was able to say, oh yeah, this was the most severe part of the fire. This would be good for collecting soil. Oh well. And then the place where I collected the soil looked like this, where there was no burning, and then I collected soil from where there had been the more severe burning. We can see the vegetation is different a year later. The ferns very typically repopulate quickly after a fire, and also the moss. So it was looking pretty much like what you'd expect for the wildfire. So it enabled us to have this hypothesis that the bacterial communities will differ in these two different soil types. And so collecting the soils proved to be a little challenging. You can see the area in this picture. There's a mixture of pine and oak trees, so coniferous and deciduous trees, and a lot of rock here, the bedrock. The soil here is generally pretty thin, generally is kind of low in nutrients. And so normally when you collect a soil sample, you dig a core, but I did rock, I couldn't dig cores. So I just collected soil from the surface. And a colleague, Eric Keeling, who, I don't know if you are familiar with the Keeling effect, is a big issue. That's his father, who, so he's a super generous person, came with me, and then he was looking at the soil, and I was asking about, this is like not what I expected. And he said, oh, that's called duff. Like duff, I've never heard of that before. So I did a little research, and duff, really the soil looked a lot like this, with partially decomposed leaves, not soil in the way that you think of typically. So this potentially plays into some of the results that we ended up with. So how we can study bacteria. We can look in the microscope, you can grow them in culture, but only about 5% of bacteria we think can be grown in the laboratory. So these molecular types of analysis that are culture independent allow us to get a broader view of what type of bacteria are present. And in molecular analysis, when you're looking at DNA, a lot of work is done with 16S PCR amplicon sequencing, gives lots of great results, and allows you to answer questions like which species of bacteria are present in this sample. Very useful. But another mechanism, the whole genome shotgun sequencing or metagenomics, can tell us which bacteria are present, but also, what are they capable of doing? What are all the genes in there for metabolism and other functions? And so just diagrammatically, when you do 16S amplicon, you only are looking at the 16S gene. You get lots of it, so you have lots of data to analyze. And this 16S gene encodes for ribosomal RNA, part of the structure of ribosomes, and is a thing that is very useful for distinguishing species of bacteria. There are additional genes to do that, but 16S is very common and popular. And then with the shotgun sequencing, we're going to sequence all of the genome. Each of these 6 genomes are chopped up kind of into smaller pieces in the sequencing, but we get the whole genome. And of course, when you have huge amounts of data like that, you need bioinformatics to be able to study that. So what I've been using for the bioinformatics is a platform called KBase. This is provided through the Department of Energy in the United States, and it's free. Anybody can use this. And it's really nice because it's GUI based. You don't have to have command line programming skills. And I find this has been really useful for undergraduate students. It allows them to think about the questions and the issues of metagenomics without having to do that coding, and then hopefully inspires them to learn the coding, because I think that's a really important skill. So what KBase can do a lot of different things, organize your metadata. You can study individual isolates through genomics, and I know the workshop in a couple of weeks is going to focus on that. You can look at RNA, build on the knowledge of what genes are present for metabolic modeling, and also these metagenomic pipelines, which is what I'll be showing you today. So it's very useful. All right, so we got soil from both burned and unburned, extracted the DNA, sent it away for sequencing, and this is the data we got. So just to clarify, I'm going to get this right one of these days, to clarify, all these genomes would be in the soil, where it's burned, there would be ashes that were left behind, and then the sequencing gives you a whole bunch of data, DNA-based sequence data. And this is what we got. Pretty good for what we paid for. We didn't have the money for huge, deep sequencing, but it was pretty happy with that data. And fairly close between the burned and unburned samples. So we could ask what types of bacteria are present initially with the reads, and then think about how does this compare to other wildfires. And so the one of the first apps in KBase, one of the first algorithms that we used is Kaiju. And this looks at the taxonomy instead with 16S, with a whole series of genes, protein genes. So what we're seeing is pretty typical in the soil that's not burned. There's largely the actinos, and then proteos, and then the acidos. That is pretty standard for what you see in soil. And then after burning, the actinos increased in number. And again, that's very common after wildfires. Proteos, there's some variability in results, but the acidos always go down. So this kind of made me feel confident in our results. It looked like other wildfires. So nice. We also looked at this at the genus level. And of course, it's a bit more data here. A lot of the bacteria, or a lot of the sequence wasn't assigned, or in this color gray, the colors are not very easy to see. There's just many types of bacteria, just a low abundance. They don't point them out. And then the highest recognizable genus was Mycobacterium. It went from almost 7% of the classified reads up to 13% after the wildfire. And this was a little surprising to me because typically mycobacteria are around 2% of the soil. So maybe our soil is a little different. Maybe our data is off, I don't know. At much lower abundance, like further down here on the chart, doesn't fit on the slide here, known pyrophilic bacteria like the arthrobacteria went from an undetectable amount to 1%, and the blastococcus also undetectable to about 1%. So again, consistent with other analyses. This was a little weird. These numbers were not what you typically see. Data is what it is. All right, so because we have the shotgun data, we could ask, do the bacteria in the unburned and burned soils have different metabolic capabilities? And what you do with these raw reads, you can put them directly in Kaiju, or you can do an assembly to try and put the pieces together into strings of DNA that overlap in their sequence into these contigs, they're called, contiguous sections of DNA. And then we can analyze the contigs for all sorts of things. And again, this is culture independence, who are potential to find out information that you wouldn't be able to do by culturing bacteria. So here's our assembly data. I use the program Metaspades, and the assembly, oh, shoot, it's in the way. What you would end up seeing here is that the burn sample has almost twice as many contigs, or twice as much base pairs. Thanks. Oh, thank you. Gracias. And in the assemblies, we had much better data in the burn samples than the unburned samples. And what I believe this has to do with is that the diversity of bacteria in the unburned sample is so much greater that the pieces of DNA that were sequenced were less likely to all come from the same genome, less likely to be able to put together. They're just like random little bits of pieces of DNA instead of these nice contigs that we would like. So of course, one of the limitations we have here is the amount of sequencing data. But we can still ask questions like, what can the bacteria at each the burned and unburned sites do? What metabolic enzymes are encoded within these two different assemblies? Okay, thank you. Let's see how that goes. So we do a couple of steps. We take the contigs from the assembly and run what's called RAST, which is an annotation program to figure out, hey, what are the proteins and RNAs and all these other functional units present in these sequences. And with the annotated genome, we can analyze these proteins. So two programs I'm going to show you that we looked at for analyzing these two assemblies are KZ, which looks at carbohydrate-focused enzymes. And our hypothesis would be that in the burned soil, because there's limited carbon, the bacteria that grow there have much more diverse abilities in breaking down carbohydrates. They have to. The easy things like glucose are not there. So they have to be more clever about it. And then microtrait looks at ecologically relevant traits. And so maybe they'll have traits in the burned assemblies that are more indicative of the environment of the burned soil. So the KZ results are it's a huge amount of data. So I just showed one little section of this. And in the K-base program, it kicks out a heat map to show you the amount in each category. And this was just a nice chunk of that data to show you what the data looks like. But when I analyze the total number of matches to the KZ enzymes in the unburned, there are 485 hits, and the burned 1610 hits. But remember that, the burned that had almost twice as much DNA. So doing the math, it still shows that there was a 16.7% increase in the number of hits. So that's consistent with the idea that the bacteria growing in the burn site have to do more in order to find nutrients in that environment. Microtraits, these are the hits that came, I actually didn't fit all of them on there, but some of the traits that came up. And so in the burned, I'm sorry, I keep hitting the wrong thing here. We see there are more of these genes that show that we can use a nitrate for producing energy, carbon monoxide way up, producing energy, and these hydrogenases that allow bacteria to use hydrogen as an energy source. And so then this too is consistent with an environment that has limited nutrients. So it looks pretty good. Okay, so then another thing we can do with the metagenomic data is take all of this and convert it back to the genomes that were in the soil. When you assemble a genome from metagenomic DNA, we call it a metagenome assembled genome, or a MAG. And so our next step was to look at these assemblies and see if we could pull MAGs out of that. This would allow us to potentially find bacteria that would never grow in the laboratory, as they're just there as DNA in the soil there. So I already mentioned we take raw sequence and assemble it, and that gives us these contigs. And the next program you use is a binning program, where it converts these various contigs, puts them together into what are likely genomes, matches them with databases to say, this contig, this contig, this contig, these would probably form an individual genome. So the bins or genomes are then analyzed for quality and further downstream analysis. So this was the data we got. I used the MetaBat for binning, and then followed that up with a program called CheckM that looks at how good the genome is, compares it to known genomes. And we see that in the unburned sample, we got very few MAGs, relatively few MAGs, and they were low quality, not a lot of green. The burned, however, we got more MAGs, and 3 of them were pretty good quality, 9, 7, and 6. Have a lot of green here. Their percent completeness calculated at 96, 96, 93 with a little bit of contamination. So only in the burn samples, and this I think speaks to the lesser diversity of bacteria in the burn samples, giving you a chance to find all of the DNA there in the sequencing and then put it back together. Whereas in the unburned sample, there's a little bit from this bacterium, a little bit from that, and little bits of each, not enough to pull together an entire genome. With this data, with these MAGs, we ran a program, GTDB, to classify each of the 3 bins. Bins 6 and 7, genome 6 was fairly big at 6 megabase pairs, 7, 4.8, more typical of Mycobacterium. And then the bin 9, smaller genome from something that's never been cultured, Pulsa 465. It's an odd name for a genus, but that's what it's called. So if you remember when I did the taxonomy, the Mycobacterium was more than we expected. And then in our mags, we're getting mycobacterium. So not surprising based on that other data, but still a little surprising based on what other research has shown. Mycobacteria are typically a small percentage of the mags that are pulled out, as well as overall data. So, hey, what are the metabolic characteristics of the mags? You have to do the RAST annotation, and then can ask, use different programs to ask about the proteins that are present. So I did a couple of quick things here with the microtraits and the KZ and I did just browsed functions in RAST. So the microtrait bioelements, what we're seeing, so bin 6, this mycobacterium, this was the larger genome. It has many more hits than the other two, but even the smaller mycobacterium genome still has a lot of hits, and this is pretty typical. Mycobacterium tend to be metabolically diverse and survive in forest environments with all that metabolic diversity. On the other hand, the smaller genome, much fewer hits. Seems to be there for more of a limited environmental role. So we see that we get varying degrees of the ability to produce energy from hydrogen. If you remember in the overall assembly, there was a lot more of this. So, you know, they partially represent what was in the assembly, but they're not representing the bacteria that presumably can use nitrate and carbon monoxide for energy. It's fine. And then this gene really popped out, different from what we saw in the overall metagenome assembly. This enzyme is known to be part of a pathway that detoxifies formaldehyde. Formaldehyde is released in wildfires, but is more volatile, so is a little surprised that there would be a selection for it within the soil, but perhaps there's a lot of formaldehyde in the soil afterwards that I haven't found data to show that, but that could be a new question to ask. So we're seeing some of the characteristics of the overall metagenome, not all of them, but that makes sense. Not every bacterium in there is going to do every function. There's a community, bacteria each doing their own thing. But it does suggest that there's some selection by the environment for these genomes. KZ, both the mycobacterium genomes have a greater diversity. And again, this is just one small part of the overall KZ analysis. This section showed a lot of differences. And again, like here, the Pulsa 465 doesn't do a lot. It seems pretty limited, but somehow it seems to be happy in this environment. So it may just be doing something very niche, very specific. Perhaps the formaldehyde is the thing it's really taking advantage of, or something else. But this gives you a sense that they're adapted potentially to this post-fire habitat. And then some of the pyrogenic organic matter that I had in an earlier slide, these aren't pulled up in the microtrait program or any other program. So I did the more painstaking thing and I just searched through the RAST annotations. How many genes are there that seem to be associated with this kind of metabolism? And yeah, there's a good amount of ability for metabolizing catechol, one of these pyrogenic organic matters. And so maybe this is another way that this Pulsa strain is doing well. It can metabolize catechol. And then these other two that I searched for, there's some ability there, a bit less. So yeah, but this supports the idea that these 3 bacteria that we pulled out as mags are happy in a post-fire environment. Okay, so I know I did rush through this a little bit because I know we started late, but look, I think we have time for this. So another thing that we ran was a Kaiju looking for viral DNA. And it's pretty diverse, but what popped out to me is this thicker blue band here, corresponding to here. Lymantria dispar multiple nucleopolyhedrovirus. Like, what is that? I never heard of it before. Of course, you know, I'm much more biased towards understanding viruses that infect bacteria. And so it increased, or I'm sorry, decreased a lot after the burning. Went from a very high percentage in the unburned soil to significantly lower, 100 times lower in the burned soil. Well, doing the research, it turns out that the Lymantria dispar is a moth called the spongy moth. I don't think it's present in South America anywhere, but in North America, it's very problematic. And in recent years, there has been kind of a large population of these moths, and the caterpillar stage eats a lot of leaves. Right? You walk under a tree and you can feel stuff dropping on you. You can hear the crunching, and yeah, it's been kind of very upsetting for people. And it turns out that the virus that's present at a high concentration in the unburned soil is the virus that infects these caterpillars. And this virus seems to be what's saving us from total loss of tree leaves every spring. The virus starts infecting the caterpillars and killing them off by quite a lot. And so it does make sense that in the burned areas, there's no leaves. There's nothing for these caterpillars to eat. So there's not going to be many caterpillars, not going to be much virus infecting the very few caterpillars. And so the amount of that viral DNA in the burned area is significantly lower. Kind of fun. That's just a picture of what this virus looks like inside of a cell. This is the polyhedron structure of it. Okay, so I hope I didn't rush through that too much, but I was trying to stay aware of the time. I will summarize. So we saw the soil microbiome at the Napanock wildfire site was similar to other viruses, forest fires, so that made us confident that the data looked pretty good. And a little surprising, Mycobacterium was more highly represented. And wondering if this is a reflection of the type of soil that we happen to study. The metagenomes and the extracted mags have genetic characteristics that are consistent with survival in burned soils. But of course, one of the big limitations that I mentioned is that the number of reads is quite small, and it limited the ability to, I think, assemble genomes from the unburned soil and even just assemble the contigs. So there might be a bias in that data. What I'd like to do in the future, I've continued to go back to that site to collect soil. I'll do so again in December or January, depending on the weather, if it's not too icy up there. And I will say that I only go in the cold weather because there's a lot of poisonous rattlesnakes in that area, and I want to wait until they are in their dens. But then, yeah, then the terrain's a little more challenging, and we see I'm a little bit of a klutz, so I have some good friends who always come on the hike with me to carry me home in case I have a fall. So that's what I think we'll be looking at in the future. And of course, none of this you do on your own. Before breaking my ankle, I did do some walking around Cerro, like this is in Cerro Concepcion, I believe, this mural that you never, you can never accomplish this all by yourself. And so how am I here? The Fulbright, the U.S. Fulbright Scholarship Scholar Program is funding me, as well as a Fulbright program in Chile. And of course, Roberto's lab. It's been so wonderful and inviting and helpful, welcoming. It's just been great. And then through KBase, they have a program called MicroNet, where they are looking to encourage more professors to use KBase in their classrooms to get more students excited about metagenomic analysis. So they paid for the sequencing and the DNA extraction kits, making that easier to do. And I am on the advisory board for MicroNet, so I'm a little bit biased. I think it's really great. Their money came from the National Science Foundation in the United States. And the students, some of their work was supported through money that's available at SUNY New Paltz for students to do research. So that is my story here. Thank you for listening. Questions?